But how does this happen? Well, the best understood example is synaptic plasticity in hippocampal neurons, and in this case, it's all to do with glutamate receptors. Glutamate is the principal excitatory neurotransmitter in the brain, and there are two particularly important glutamate receptors: AMPA receptors and NMDA receptors. The AMPA receptor is permeable to potassium and sodium, and it is this inward flux through the AMPA receptor which depolarizes the cell. The NMDA receptors, in contrast, are blocked by magnesium at negative voltages, and therefore do not significantly contribute to postsynaptic depolarization of the cell. However, once the cell is depolarized, the magnesium is displaced, and ions then flow through the NMDA receptor. Importantly, the NMDA receptor also allows calcium to flow through. It is the nature of this calcium current which causes spike timing dependent plasticity. If the presynaptic neuron fires first, it becomes depolarized and releases glutamate. The glutamate binds to AMPA receptors at the postsynaptic neuron, causing it to depolarize. As the cell becomes depolarized, NMDA receptors become unblocked. Glutamate binds to them, and this causes a large calcium influx. If the postsynaptic neuron fires first, it becomes depolarized. As it is repolarizing, the presynaptic neuron fires and releases glutamate. When the glutamate reaches the postsynaptic cell, it finds it repolarizing and at a lower voltage. This means there are fewer NMDA receptors available to bind to. This leads to a more moderate calcium influx. A large calcium influx leads to long-term potentiation, or LTP, and a small calcium influx leads to long-term depression, known as LTD. But how does this happen?